Welcome everyone. First of all, I would like to thank Thermo Fisher for inviting me here so that I can show you some of the work that we did at IPATIMUP, where we did test the Genexus using a custom AmpliSeq, in this particular case for testing the RASopathies. These are the disclaimers. I should also add that currently I'm a Thermo Fisher employee, but all this work was done previous to my integration in the company. All the work that I will present was done and developed at IPATIMUP. IPATIMUP is a funding member of I3S, which currently is the leading research institute in the topic of health. Within the institution, we do have a diagnostic facility where the mission is actually to perform genetic diagnosis and parentage testing to the society in Portugal. But one of our key missions there is to have good quality of our testings, and this is mostly shown by our different laboratory accreditations. That goes from CAP accreditation to the ISO 15189 and the ISO 17025. As I said, what I will be discussing today are the RASopathies. And the RASopathies are a group of rare genetic conditions caused by mutations in a growing number of genes in the RAS-MAP kinase pathway. And while these individual syndromes are rare, as a group, they are amongst the most common genetic conditions in the world. The syndromes have a lot of overlapping phenotypic features, which makes relevant for the identification of the genetic underlying genetic defect. As such, the diagnosis of these disorders is particularly difficult to perform both in utero and at first years of life, as most of the features are late during childhood that actually present themselves. Consequently, the molecular analysis has become an important key for the verification of the clinical diagnosis and represents a highly informative prognostic tool. We at the Institute have been actually in the forefront already in 2012 of moving our genetic tests into next generation sequencing strategy. This was in the early days with the Ion PGM and the IOOT2, which meant that was actually a quite highly manual procedure, but in a way allowed us to make it a reduction in our turnaround time that by then was around 3 months to produce the test to close to 1.5 months. Importantly was also that the increasing number of genes associated with these disorders became more and more prevalent, so that we had to increase the number of allowed us to increase the number of genes that were included in the tests. With time, we sort of increased our capacity to automate the test, in parallel also increasing the number of genes that were being included. This allowed us to reduce the turnaround time to close to, let's say, 15 days. Automation was not only at the stage of library preparation, but mostly at the templating stage. And the introduction of the Ion S5XL allowed us to have a much quicker response in terms of sequencing, and also the introduction of the automation for bioinformatic analysis allowed us a more simplified testing strategy. What was interesting for us was that in the early mid-2019, we had the opportunity to get the Ion Genexus sequencer at the institution. And there we had the opportunity to, at the first hand, do a complete test of the system. And one of these tests was actually using the RASopathies panel that we had recently developed. This panel is a custom Ion AmpliSeq design. which we set it to have an amplicon size not very large, that could go up to 275 base pairs. It had to be divided into 2 pools, and this custom panel allowed us to increase the number of genes that we introduced in the test, which is currently now 23 genes. Some of the genes are clearly important for the development of these different syndromes. Some others are less relevant, but with the capacities that we have nowadays for next generation sequencing, this was an opportunity to expand the test. What we did is, in this very pilot study using the Genexus, was to include a few, in this particular case, 6 control samples. And what I mean by control samples is that are samples that have been previously used for clinical research, so that we had complete genetic characterization of these samples. Within these 6 samples, we tested 3 that were positive, 3 that had no clear pathogenic variant, and also, very importantly, we included samples that were prenatal testing samples and also postnatal testing samples. And the postnatal testing samples were both from very early age to a later age of the individuals. So this is actually the first run that we did using this particular test. And what one can see from this snapshot of the Genexus screen is that we only had to use a single lane. So we didn't have to run the entire ship to do this test. We actually just used a single lane. This single lane produced the expected number of final reads, that is between 13 to 15,000 million reads. In this particular case, it was 13 million reads. The average size of the fragments is at the level that was expected, very close to the 200 base pairs, and all this with a mean accuracy extremely high, close to 99% of mean raw accuracy. What we wanted then to do was to use as I said before, these samples had already been characterized. So now we wanted to do a head-to-head comparison towards the results that we had previously obtained. The first thing that we looked was at more of a QC perspective. So we compared the uniformity of amplicon coverage, the percentage base reads on target, and then the uniformity of base coverage between the run on the Genexus and the run on the Ion S5. And we divided the samples due to their particularities in the quality and the quantity on the postnatal clinical research samples and the prenatal clinical research samples. And what we can see from this plot here is that the results are extremely comparable in coverage analysis between the 2 systems, and showing us that the results most likely would be very comparable. What we then next was now to look at the concordance between the 2 systems on the variant calling. And this is just a simple comparison of actually 2 of the samples, in this particular case 2 of the positive samples, one prenatal clinical research sample and the other postnatal clinical research sample. And what we see here is then the comparison between the Genexus variant calling and the S5 plus Ion Reporter variant calling. And what we see is that the exactly same variants were detected between the two systems and the 2 samples. And importantly, the allele coverage and more relevant, the allele ratio is highly comparable, meaning that the 2 systems are actually sequencing at the same high standards. Furthermore, then we did reproducibility tests where we did a second run using the exact same samples and running on a different lane of the actually the same chip. And what we did then was to compare how the reproducibility was between the two different runs on the Genexus. And not to our surprise, it came with a very high level of reproducibility, here seen by both the qual score, the coverage, that is very similar between the two runs, and again the allele ratio between the two runs and the different genotypes of the sample. So, making it a very quick group of conclusions from this very simple pilot study, we can show that the results are highly concordant between what was our previous standard of Ion Chef plus Ion S5 sequencer plus Ion Reporter bioinformatic analysis and the all-in-one Genexus sequencer. We show that the assay, the custom AmpliSeq assay, works very well both for DNA from the amniotic fluid and recovered from whole blood. Our calculations set that around 16 samples can be run in a single lane of the Genexus with adequate base coverage so that variant calling can be performed successfully. But what is relevant is that in this particular case, by using the Ion Genexus, we can have a runtime from sample to result with within less than 16 hours, which in particular for the prenatal setting of genetic testing, this might be of extreme relevance. So overall, using a custom AmpliSeq gene panel can be used in combination with the Ion Genexus sequences for both rapid, flexible, and fully automated research testing. Of course, this was not done just by myself. There were a group of people involved, both at IPATIMUP and also at Thermo Fisher. And with this, I thank you for your attention.
And thank you very much for this first presentation. Thank you very much, José. Very interesting to look at the application around the brand new Ion Genexus system. Now at this stage, I would like to open the Q&A. So I'm going to check for questions from the participants around here. So the first question, José, for you will be, how much time did it took to you and your team to actually establish the new workflow? So once you got the instrument in place, how long did it take, to have it all set it up and say, okay, this is now ready, up and running?
Thank you very much for the information and also for the invitation to be here. I have to say that everything was quite fast. The learning curve it's really simple, so you really have almost no time needed to get acquainted with the system. Everything is automated. All what had some time to learn was mostly on how to look at the data in the end, because it's a new software version, so you need to learn where to look and where to click. But for sample preparation, since you only have to include DNA into the system, that was almost no learning curve. Then all the system is done very much automated. There's a vision system that controls all the reagents that one needs to set into the machine. So it's in a way almost childproof, because the system warns you if you are placing the wrong tube or a tube in the wrong place or any of the reagents. So it's really easy to learn.
Okay. Thank you very much for that. We actually have time for only one more question. And the question is about, you use this instrument for this specific application. Do you know, or can you share what's the plan for IPATIMUP if they are thinking about expand the usage, or they will stay on these? Does anything that you can comment on that?
Yeah, sure. So the system at the Institute most likely will not be a dedicated system for a given pathology, for say, like for example, not for this, in this particular case, for RASopathies. What I might see in the future is that this system, so we do still have all the other sequencers. So those sequencers will be kept as our sort of working horses. So most routine testing that are performed in bulk, let's say, will keep on going on those systems. And I see this system, due to this particularity of speed, to be used as a fast track. So every sample that comes with a certain urgency, either because of diagnostic or prognostic or predictive test that needs to be performed, it will most likely go into that fast track.
Thank you very much. So it seems like really speed is one of the most relevant new feature of this instrument. Hopefully there's something more. Now, with no further delay, I would like to move on to the second speaker for today. So once again, thank you very much, Carl, for being with us. The scene is all yours. Looking forward for your presentation.